KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Good morning students. Now in this video we are discussing electron transport. So electron transport system, so how it will be taking place means as we have seen before. So to explain the transport of electron, so there are two methods over there. One is Z scheme. Z scheme means it is also known as non-cyclic electron transport. One is non-cyclic means definitely second one will be cyclic. Okay, so that is the information. Now we will start to discuss. So before start the information of non-cyclic electron transport, first of all one cyclic form I will give you. So that is the thing. Now first let us observe the non-cyclic electron transport. Non-cyclic electron transport. Later, what is the information we are discussing now? So that information, so here let us see. First of all, what is happening now? As I told you before and as we have learned before, light reaction which occurs on the membrane of the thylakoids. Or simply thylakoid membranes. So thereby why sir light reaction occurs in the thylakoid membrane means one more point we know very well. On the thylakoid membranes there are different types of pigments are there. Photosynthetic pigments in previous video we learned no chlorophyll A, chlorophyll B, carotenoids, xanthophylls likewise. So there are various types of photosynthetic pigments which are present on the thylakoid membrane. So now because of the presence of pigments on the thylakoid membrane, what is happening? Absorbs the sunlight. Sunlight, light, simply so light. So now it is coming towards the light harvesting complex first. So when the light harvesting complex receives the light now, at the time what is happening now? One point marandavenda adenana. So the entire non-cyclic electron transport processes which occurring on the thylakoid membrane. Membrane is there no on the thylakoid. Yesterday in chloroplast we see no the group of thylakoids are known as grana. So each and every thylakoid so like membrane is there. So that on the membrane we will notice the presence of pigments. So these pigments have the capability to absorb light. So that is only here antenna formation takes place. Antenna means here antenna involves in the formation of chlorophyll B, carotenoids, xanthophylls, and antenna later reaction center which is formed by the chlorophyll A alone. So did you remember that information? Likewise, so one light harvesting complex is there. This light harvesting complex receives the light. So thereby on the thylakoid membrane water in the presence of light is going to be splitted. It is going to be splitted. So thereby what is happening means, so what is happening, two electrons and two protons and of O2 formation takes place. Suppose here if you keep two, so if you keep two molecules of water, here complete oxygen molecule formation takes place. Okay, but here what is the point here from this reaction we can understand. Suppose if you are taking one molecules of water now, here half O2 only formation takes place. Later 2 electrons plus 2 protons will be released. Okay, what is the information? What is the information now here we can say when the splitting of water process taking place. So if you are going for neat information, deep information if you take chapter mineral nutrition, the major and the principal function of manganese we see no, it involves in the evolution of oxygen or the splitting of water. Did you remember? Or photolysis of water. Did you remember these terms? So that is the point here only taking place, means on the thylakoid membrane. So here splitting of water process is taking place, and here the electron transport chain, means here the splitting of water which occurs within the thylakoid membrane, and the electrons are produced, and once the electrons are liberated by the splitting of water, no, that comes, or the electrons are enters into the so electrons are received by the first electron acceptor. Now here the first light is accepted by the PS2, okay, photosystem 2. So here, that's why here I have denoted PS2, here it will be there. So that first of all, what are the electrons or which are coming by the splitting of water received by the PS2, means P680 nanometers, means maximum wavelength of light is P680 nanometers. Later the 2 electrons, here 2 also we can write 2 electrons which are received by the first electron acceptor phi of 18. So while explaining the neat information, so we will have that information, no worry. So here electron acceptor phi of 18, so that's why here we have denoted PS2. So from the electron acceptor to the, now the electron transport is going to be started, or electron transport system. Through the electron transport system, later 2 molecules of or 2 electrons which are conveyed to the photosystem 1. So that's why here we have written photosystem 1. So later from the again photosystem 1 to by again with the help of light. So here one more time here we can write light. Again 2 electrons are excited, and here what are the electrons which are coming from the PS1 received by the, and one more electron acceptor. Okay, here the one more electron acceptor are FAS protein. Okay, FES protein. So that is also very exclusively we will have in deep studies of NEET exam. Later here this entire information that we can be taken under PS1. So later what are the electrons or which are coming from the PS1 and enters into the PS2 or received by the PS2. Later, so these electrons which are involved in the formation of NADPH, means here the reduction reactions are going on. Okay, reduction reactions means we know very well, addition of hydrogens or the removal of oxygen. So now NADP NADP plus, now it is converted into NADPH. So in this particular case, no. So finally in non-cyclic electron transport system, if you see there, no, here in electron transport system, ATP molecules, means here the ATP is formed from the adenosine diphosphate, means this ADP, ADP plus PI. It is not appearing clearly, but through my voice you can save ADP plus PI. PI means inorganic phosphate. So now ADP path adenosine diphosphate, and pakatali one inorganic phosphate is there. Diphosphates ange rendu ange in one, totally 3 ATPs or 3 phosphates structure formation takes place. So that is only adenosine tri means 3 only, no, triphosphate. Okay, so now here one ATP formation we are observing, and later one NADPH formation also we are noticing in Z scheme. Z scheme is nothing but here non-cyclic electron transport. Ipu one important point ipu in the presence of light or with the help of light, ATP formation we are noticing, or we are studying the formation of ATP. So thereby, suppose in presence of light, suppose ATP formation takes place means, suppose if ATP formation takes place means, so such kind of a process we are calling as photophosphorylation. One more time you see, photophosphorylation. One point we learned where in discussing of the role of macronutrients, means CHO, NPK, CAMGS, while discussing about that one. So even the phosphate while discussing about the phosphate, we have discussed phosphate which helps in phosphorylation reactions we see. Up to one point we learned what it is. Phosphorylation reactions are nothing but formation of ATP, or the phosphorylation processes are nothing but the processes in which ATP formation takes place. Okay, so now here phosphorylation means we know very well ATP formation. Now formation of ATP taking place with the presence of light here. So thereby this particular ATP formation we are considering as photophosphorylation. Photo means light. So in the presence of light ATP formation is taking place or is going on. That's why this entire reaction or the formation of ATP or processes we will be taking as photophosphorylation. Suppose if there is no involvement of light but ATP are formed means at the time photo we should not add. Photo suppose here light involvement is there in the formation of ATP means then only we have to add photo. Suppose there is no involvement of light means but ATP is going to be formed means at the time what we have to pronounce? Photophosphorylation. I hope you understood. Now here, sir, then why we have given a name to this particular flow chart, why we have given a name as non-cyclic electron transport means non-cyclic means, so where the electrons are started, so from there to the electrons are moving and the electrons are not coming to the origin place or not coming back where the electrons are originated. Okay, so means here and one more place, and then why we have given a name as a jet scheme means idu paathumbodhu paru. So electrons first of all started to move from the LHC means light harvesting complex to, so from 2 to 1 and from 1 to again involves in the formation of NADPH means here uphill transport and downhill again uphill. So here idu paathumbodhu actually seems to be N madri, but scientists they have given a name it as jet scheme. I think so they have seen like this. So here paathumbodhu it is a Z, it means if you turn it no, so that will be Z no, jet scheme mechanism or jet scheme electron transport chain. Ipo inge rendu points remember pannikonga. Adu enna in non cyclic electron transport there is a split of water in the presence of light. Means here the electrons as well as protons which are liberated from the water. That's why in introduction class of photosynthesis we discussed in green plants what it is, hydrogen which is liberated from the or liberated by the water molecule, but in case of bacteria, so hydrogen which is liberated from the H2S. Hydrogen sulfide we discussed. Did you remember it? So likewise that is one important point. And in non-cyclic electron transport ATP formation as well as NADPH formation taking place. So thereby did you remember one point, this ATP as well as NADPH once we have called them as assimilatory power. So and one more point, ATP as well as NADPH we are considering as a products of light reaction. So these two products of light only, so definitely involves in the process of dark reaction which occurs in the stroma of chloroplast. Okay, I hope you understood this one. Now based on this information, now let us discuss the what is the information is present in jet scheme or non-cyclic electron transport. The movement of electrons is downhill in terms of an oxidation and reduction or redox potential scale. So the electrons are not used up as they passed through the electron transport chain, but are passed on to the pigments onto the pigments photosystem 1. So means here definitely what are the electrons which are liberated at the by the splitting of water. So all these electrons are transported continuously or the translocated continuously from the PS2 to PS1 and finally reach to the final electron acceptor. There the electrons which are involved in the formation of NADPH. So now here simultaneously electrons in the redox center of PS1 are also excited when they receive the red light wavelength. So P700 nanometers. So here P700 nanometers, and transfer to another electron acceptor molecule, so that has a greater redox potential. So thereby here what is the thing here in a non-cyclic electron transport flow chart we have seen. First of all PS2 we seen. So there the maximum wavelength of light is P680 nanometers, and in PS1 maximum wavelength of light is P700 nanometers. So likewise, so when the light is going on, or when the light or with the presence of light, so definitely the electrons which are transferred from the one place to another place without any rest. So later in addition of these electrons reduces, so here addition of these electrons reduces the NADP plus to NADPH plus. Already we have seen. Finally this particular product is formed so by the addition of the electrons to the NADP plus. Next one, this whole scheme of transfer of electrons starting from the PS2 already we have seen clearly. So means that particular whole scheme if you are going to see. So from where the what is the origin place of the electrons means, so that one kandipa we can say without any doubt the location is PS1. Next one, excitation or the excitation of electrons transfer to another acceptor, and finally downhill to NADP plus, reducing it into NADPH plus again. So means here what is the point? What are the electrons which are started at the PS1? So finally first of all electrons are received by the first electron acceptor, later electrons are passing to the PS1, and from the PS1 to again electrons are excited and received by the final electron acceptor, and finally electrons are involved in the redox reaction. So thereby your NADP plus are reduced to NADPH plus H plus. That's why it is called as Z scheme. Due to its characteristic shape, the shape is formed when all the carriers is placed in a sequence on the redox potential scale. So by seeing that all carriers of the electrons, so by seeing the carriers, so it is seems to be Z flow of electrons or Z-shaped flow of electrons we are noticing. So that's why this particular flow of electrons we are calling as Z scheme electron transport. Now, so here this particular side heading is a backbone. This particular side heading is a backbone to the starting the process of non-cyclic electron transport. So what is a backbone process? Splitting of water. First splitting of water process only should be taken place. Suppose if the splitting of water or the photolysis of water process is not going place, how non-cyclic electron transport chain will be started? No chance, isn't it? So thereby first of all backbone process to the non-cyclic electron transport is splitting of water. So at the time the electrons that were removed from the PS2 must be replaced, and this is achieved by the electrons available due to the splitting of water. So means here if you see there the electrons which are moving from the PS2 to, and it is moving from the are moving towards the PS1. So one more time, the electrons which are moving from the PS2 to PS2 to PS1 at the time. So finally the electrons which are received by the PS1 and the electrons later received by the last electron acceptor, later redox reaction. So thereby definitely NADPH formation is we are observing. Later again what are the electrons or how these electrons so reach it to the PS1 sir means again, so one more water molecule must be splitted, later again electrons are conveyed to the PS2, later again electrons are conveyed to the PS1. So likewise, so definitely each and every time one water molecule so should be splitted with the help of light. So that's why what are the electrons which are carried, or what are the electrons that were removed from the PS2 again replaced at the PS2 only by the splitting of water. Emma, one more time let me explain. Ipo splitting of water means we know very well. Splitting means separation of water. Adi eppadi separate avana? So one molecule of water, suppose if you see there now, that will be converted into 2 protons and 2 electrons plus of O2 formation takes place, isn't it? Ipo what is happening in the presence of light? In the presence of light, one water molecule is going to be splitted, so thereby 2 electrons are transferred to the photosystem 2. Again, if one point from the photosystem 2 to the electrons are moving to the photosystem 1. If there is no electrons in the photosystem 2, isn't it? Why? What are the 2 electrons are present in the photosystem 2 that are translocated towards the, or that are transported towards the photosystem 1? If there is no electrons in the photosystem 2, again now electrons will reach to the photosystem 2 only from the splitting of water. Means here PS2 getting of 2 electrons means that is only accomplished or achieved by the splitting of water only. Means definitely 2 electrons are moving towards the PS2. But here what is the point we have to remember? What are the electrons are there at the PS2? These 2 electrons are coming because of the splitting of water. Again, the what are the electrons are present in the PS2 definitely should transfer or should move towards the PS1. Again, if PS2 nalli there is no electrons, again splitting of water process takes place. Again 2 electrons which are moving towards the PS2. Again from the PS2 to electrons are moving towards the PS1. So likewise each and every sequence, each and every time there is a splitting of water and 2 electrons are produced, and electrons are again finally moving towards the PS1, and from there to redox reaction and involves in the formation of NADPH. So when this particular process is going to be taken place, so at electron transport system even ATP molecules also formation takes place. So thereby what is the point? In a non-cyclic electron transport, 2 points you don't forget. So one is there is a involvement of 2 photosystems. One is of photosystem 2 and one more is photosystem 1. And the next point is, so here in a non-cyclic electron transport there is a splitting of water molecule with the presence of light. And one more point here, there are 2 different types of assimilatory powers. One is ATP and one more is NADPH is produced. These 3 points marandavenda. Okay, next one, let us continue to see the splitting of water information. Now the splitting of water is associated with the PS2. Already we seen associated with the PS2. Now water is split into 2 protons plus of O2 and electrons. Next one, this creates oxygen, one of the net products of photosynthesis. So thereby here what we can get the point? So in the process of photosynthesis, now we are obtaining or we are observing the formation of oxygen as one of the net products. So thereby when the process of photosynthesis is going to be taken place, definitely it is a confirmed to produce the oxygen as one of the net products. So that's why in earlier experiments, John Ingenhouse, when he was performed one experiment by taking off aquatic plant hydrilla, he was observed the small bubbles when he was placed the particular hydrilla plant in water in presence of light, and he was observed the formation of water bubbles around the plant. Did you remember that? Likewise here also definitely oxygen is liberated in the process of photosynthesis. Next one, the electrons needed to replace those removed from the PS1 are provided by the PS2. Means first of all PS2 will supply the electrons to the PS1, and from the PS1 to helps in the redox reactions to form the NADPH. Again now here PS1 and PS2 also not having electrons. Why? Because what are the electrons first are gone, are involved in the formation of NADPH. Again water splitting process takes place. Again electrons are moving towards the PS2, aprom PS1 ku povum, aprom NADPH formation ki involve. So likewise, so each and every time the process which will be continued furtherly. So that's why here what is the point? So if you take 2 molecules of water, 4 protons and 1 molecule of O2 ikke para, one complete oxygen molecule formation taking place and 4 electrons. Okay, so this is a point mainly we have to focus. So suppose here if you take only 1 molecule of water, no, there is no chance to the formation of one complete oxygen molecule. Half matin the formation now. So that's why to form the one complete oxygen molecule here, we took 2 molecules of water. If you take 2 molecules of water, kandipa ange 4 protons and 4 electrons formation we can notice. Okay, so next one we need to emphasize here that the water splitting or the splitting of water complex is associated with the PS2 which itself is a physically located on the inner side of the membrane of the thylakoid. So thereby here the splitting of water, the process already we have seen. So thereby the splitting of water process which occurs inside of the thylakoid membrane. So this is a brief information so which I can give related to the non-cyclic electron transport, but I will promise you while explaining the neat information. So this entire information, so I will explain even by taking the structure of thylakoid membrane. On the thylakoid membrane, this entire structure drop panni aprom neat learn panikalam. Okay ma, so this is just it is a textbook diagram, but a neat information diagram appo idu koncham deep arika that time, so let us learn deeply, no problem. So now also what are the maximum points are required we have discussed even this particular diagram. Okay, now second one is cyclic photophosphorylation. What it is? Cyclic photophosphorylation. Ipo paatha cyclic means what are the electrons or where the electrons are started, again finally the electrons are reaching to the same located point or the same origin point. That's why we have given a name it as cyclic. Ipo benzene paatham boda. So when we are going to draw one particular benzene structure, no, that one we will be taking as a cyclic form. Why? Because they are all C6H6 only, no, so there the particular structure will be like closed structure, no, adidana cyclic. So like that here also the electrons are started to move at the PS1 here. PS1 is nothing but P700 nanometers only, no. So electrons are started here and the electrons are moving received by the first electron acceptor. Later the electrons are involved in the electron transport system and again finally the electrons are reaching to the origin location or the starting point. So that's why ipo paatha, so inga vandachu. So likewise the electrons are cycled. So that's why here, but here even here also, so there is a byproduct that is ATP. So means that's why here what is the name here we have given photophosphorylation, means here also in the presence of light there is a production of ATP. That's why suppose here light involvement not there, means just we will put side heading only phosphorylation, but here light involvement also is there, no, that's how we are going to take it as photophosphorylation. Here also electrons which are coming from the light, so coming from the light and falls on the chlorophyll photosystem 1 and excited and later received by the electron acceptor, later moving towards the electron transport chain and again coming back to the origin place. But when the electrons are moving in a cycle manner, no, so there also we will notice the formation of ATP. But my dear students, inge one point nalla save paniko, here in a cyclic photophosphorylation only we are observing the formation of ATP, there is no chance to the formation of NADPH. So this is a main important point that we can learn from the cyclic photophosphorylation. So this is the information is enough for the board examination. So while discussing about the neat information, so in which kind of organisms photophosphorylation process takes place, and so who is the name of the scientist that discovered the photophosphorylation, everything in detail let us learn. Okay, so this is the information is enough for the primary learning. So if you understand this one, later let us go in a deep manner. I hope you understood this. One or two times பார்த்துக்கோங்கம்மா. Suppose அங்கே புரியலைன்னா I am there, no worry. So when you will come to the school, definitely. So suppose if you would like to get the information from the entire textbook also, no problem. I am ready to explain. Thank you and be safe at home.